Dear students, Assalamu alaikum wa rahmatullahi wa barakatuhu. Welcome back to AmreenaMehrajTutorials. I hope you remember that in last lecture we have started genetics for class 12. In my last lecture I have made you understand what is mutation. I have given you classification of mutation where we have seen there are two types of mutations, either gene mutation or chromosomal mutation. In last lecture we have completed gene mutations whereby I have made you understand what is basically gene mutation, what are the two types of gene mutations, rather three types we have. We have substitution, deletion and insertion. I made you understand about frameshift mutation also in my last lecture. Now let us go to chromosomal mutations. I would like to request that if you haven't subscribed my channel yet, do subscribe, like, share and comment, and don't forget to press the bell icon if you want to get notified with my videos. Let's get started with the chromosomal mutations. Chromosomal mutations are, my dear students, those mutations which happen to the chromosomes. That means either you will find the number of the chromosomes increasing or you will find the number of the chromosomes decreasing in this particular case. Even you will find certain changes whereby you will see that the structure of the chromosome is getting changed. So chromosomal mutations, accordingly, you can classify them into two types. Number one type where you will see numeric change happening to the chromosomes, numerical change in chromosomes. I hope you remember numerical change means the number of the chromosome is changing, either decreasing or increasing. That is the secondary question. Or you have what? You have the structural change happening to the chromosome. So now we are not concerned with the gene, we are concerned with the entire chromosome. We are concerned with chromosome. So structural change happening to the chromosome or numerical change, structural change in chromosomes. So numerical changes, you call these changes, you use the term genomic mutations for these, and you use the word chromosomal aberrations for these. Okay? So whenever you have some change happening to the number of chromosome, it's called genomic mutation. Whenever you have some change happening to the structure of the chromosome, you call it chromosomal aberrations. We'll be taking this entirely in next lecture. Let's remain focused to the numerical change in chromosomes. As far as numerical change, my dear students, are concerned, again you will be having two types of numerical changes, as I told you. Either you will see the number of the chromosome increasing or you will see the number of chromosome decreasing. So accordingly you will have two cases. Number one is called aneuploidy. Pay attention. And number two is called, I'll make it short because I need that space. Number two is called euploidy. Euploidy is also called, it's also called polyploidy. Okay? Pay attention. Aneuploidy is the case, my dear students, where you will see that the change in the number of chromosomes is happening in one or few numbers. So either you have one or few chromosomes added or deleted from a set of chromosomes. Pay attention. Like you have either one chromosome reduced or one chromosome added. You have two chromosomes reduced, two chromosome added to the entire set of chromosomes. Say for example, if you are talking about humans, you will be having either, normally we have 46 chromosomes in our cells, either you will be having 47 chromosomes, 48 chromosomes in this case. Or you will be having, say for example, 45 chromosomes, 44 chromosomes. If that is the case, that means what? One chromosome is being added or one chromosome is being deleted. So there is basically what? There is loss or gain of one or few chromosomes. You can put it in this way also. Aneuploidy is that type of numeric change in chromosomes, that type of genomic mutation in which you have loss or gain of one or few chromosomes, to be very simple. Okay. Now, as far as euploidy is concerned, it is the same type of mutation, but you will not be having one or two chromosomes added. you will be having a complete new set of chromosomes. So in this case, you will not find only 1 or 2 chromosomes added. You will see that entire haploid set of chromosome is added. So in this case, you will see an entire haploid set of chromosomes is added to the genome of an organism. Suppose an organism is, say for example, a normal organism is diploid. So he or she is having what? He or she is having 2 sets of chromosomes. But if the same person will have now 3 sets, means what? Means what? Entire set is increasing. You have a new haploid set. The condition has become 3n. Then you will call it polyploidy or euploidy. If the condition is like an organism is not having 3 sets, an organism is having 4 sets of chromosomes. So entire set. So here you are concerned with what? You are not concerned with 1 or 2. You are concerned with a complete set of chromosomes. So this is the basic thing, okay? Now, what is basically happening? What is the reason for this increase or decrease in the number of chromosomes? That is very important. Once you will understand that, then I will go further into aneuploidy and euploidy. Let me make you understand first of all, where is this reason for polyploidy? For that matter, let me make you understand. My dear students, you might be knowing this thing. This is very basic. Try and understand. Pay attention. I'll be giving you what? I'll be now giving you a reason for this aneuploidy specifically. Rather, you can get the reason behind the numerical change in chromosomes. The reason lies in this. Reason lies in cell division. Okay? There are 2 possible ways of cell division. You have mitosis or meiosis. That's clear to you people. Now, it's some students have this idea that these mutations happen when you have some error in meiosis also. Let us get out of this confusion. It's not always meiosis, being improper that results in aneuploidy or euploidy. Sometimes there could be defect with mitosis also. So that's why I am not pinpointing anything. I'm saying the reason lies with fault in cell division. Okay, so it can happen both in case of faulty mitosis or faulty meiosis. Now let us we will ponder more upon meiosis. So if you know about this meiosis, this type of cell division is taking place when what is formed? When gametes are formed. Okay, so if something wrong will happen to your meiosis, then there is a chance, not only a chance, there is 100% assurance that the gamete which will be produced will not be a normal gamete. It may be an incorrect gamete. Okay, pay attention. When you talk about meiosis, my dear students, initially when we talk about meiosis 1, okay, at that stage we have a cell which is having homologous pairs of chromosomes. Okay, so suppose I'll take over here chromosome number 1 into consideration. Okay, I'll draw chromosome number 1. So you have how many chromosomes present in chromosome number 1? You have 2 chromosomes. Okay, so suppose this, which is on the board right now within a cell, this represents your chromosome number 1. What is it? It is a homologous pair. Actually, you have understood this in class 11th meiosis, but let me make you understand over here once again in nutshell. So you have chromosome number 1, it's homologous pair. We are talking about meiosis 1. So you have chromosome number 2, homologous pair, chromosome number 3, homologous pair. Why I use different colors? Because I want you people to get an idea this organism has having chromosome number 1. In that pair, 1 chromosome has come from the maternal side, 1 chromosome has come from the paternal side to this particular organism. Okay, so you have this pair. Very simple. You have pair of chromosome number 1. Similarly, you have pair of chromosome number 2. Similarly, you have pair of chromosome number 3. I'm making it easy. Similarly, you have what? Chromosome number 2 pair. Similarly, you have chromosome number 3 pair. So suppose how many pairs are right now on the board? Chromosome number 1 pair, chromosome number 2 pair, chromosome number 3 pair. Okay. So whatever I'll be showing happening to this, that is happening to the entire homologous pairs. Okay. So I'll remove these. Let us try and understand. when meiosis 1 happens, before this particular cell division, before particular meiosis, what has happened is basically DNA replication. So you have this copy, replication is already taking place, okay? So you have this in the form of a copy. So each chromosome is having how many chromatids? 2. Okay, very good. So this is basic thing because the application has already happened. Now what happens during meiosis 1, specifically when we talk about anaphase 1? Think what happens in anaphase 1. What is the job of anaphase 1? During anaphase 1, this homologous pair of chromosome has to undergo what? It has to undergo a separation. Means it has to separate. Out of this pair, out of this homologous pair, one chromosome will go to one cell, another chromosome will go to another cell. This is called disjunction, if you remember. Okay, this is called disjunction. So in normal sequence, normal meiosis, what will you see happening in anaphase 1? You will see that during anaphase 1, this will separate like this, and you will have 2 cells. You will have 2 cells. Now, each cell, this particular cell will be having this chromosome, and this particular cell will be having this chromosome. So what has happened to the chromosome number 1? Separation of chromosome number 1 has taken place. Separation of, rather we must be specific, separation of the homologous pair of chromosome number 1 has taken place. Okay. Now in meiosis second, further when the meiosis second will complete, you will have out of this, you will have finally how many cells? 4 cells you will have like this. Okay. So this cell will have 1 chromosome number 1. This will have 1 copy of chromosome number 1. This will have 1 copy of chromosome number 1. And this will have 1 copy of chromosome number 1. Why I am using chromosome number 1? Because I have taken into reference chromosome number 1. The same thing is happening to each and every 23 pair of chromosomes. This is your normal meiosis. So this is happening in your meiosis second. So finally you have how many cells? Rather you say you have 4 gametes at the end. Each gamete is what? Haploid, and each gamete is having only 1 copy of the parental chromosome. That must be clear. Now my dear students, think, if this disjunction will not happen, if this disjunction will not take place, if this separation will not take place, think about this. If the separation will not take place during anaphase 1, that's why I said reason lies in cell division. Meiosis mein gadbad hai. So reason lies with meiosis. So suppose the separation has not taken place, remove this line, no separation has taken place. Say for example, both of these chromosomes get shifted to one cell, okay? So this cell received nothing, okay? This cell didn't receive anything, and this cell, on the other hand, this one, which is on the left side, it received this chromosome also. So that means chromosome number 1, both of its copies went to the same cell, okay? Now finally, after meiosis 1, you will see that these 2 cells will totally be lagging in chromosome number 1. Chromosome number 1 will be absent in them, okay? And what will be the case with these 2 cells? Think it's very tricky. The case will become like this at the end. You will be having 2 cells like this. This cell will be having this cell will be having 1 copy of chromosome number 1, and the same cell will be having another copy of chromosome number 1. So I will have 2 copies of chromosome number 1. This is also having what? 2 copies of chromosome number 1. Okay, so here you have 2 copies of chromosome number 1. Here you have 2 copies of chromosome number 1. This gamete is not having chromosome number 1. This gamete is not having chromosome number 1. What could be the possibility, my dear students? What could be the possibility? The possibility is, you need to be very smart. Try to think in a smart manner. Say for example, the gamete which I am talking about right now, this gamete is basically an ova. Say for example, it's an egg, okay? Suppose it fertilized with a sperm. This is a sperm, the normal sperm which is having 22 autosomes, and it's having suppose Y sex chromosome. Okay, suppose these two fertilize with each other. You have a zygote. Now zygote, the condition of zygote will be what? This is having, suppose, X chromosome. Okay, so you have X and Y. X and Y. Done. So the children will be what? Male. But this is having how many autosomes? 22. This is having, rest of the autosomes will be correct, but chromosome number 1 will be present in how many number? 2. Okay, so this particular gamete is not having the normal 22 autosomes. It's having 23 autosomes. Think why it is having 23 autosomes? Because it's having one extra chromosome number 1 copy. So it has 2 copies of chromosome number 1. Rest of the chromosomes have only 1 copy. But chromosome number 1 is present in 2 copies. So total autosomes in this particular cell will be 23. So this cell is having 23 autosomes. This gamete is having 22 autosomes. What will be the total number of autosomes? 23 plus 22 is how much? It's 45 autosomes. So we'll be having 45 plus XY. What is the total number of chromosomes? 45, 46, 47. So it's having, this organism is having how many total chromosomes? 47. Is it normal? No. A normal organism must have how many chromosomes? A normal organism must have 46 chromosomes. But over here you are having 47. What is the reason? The reason is this extra chromosome, this chromosome number 1 in 2 copies. Because of what? Because of this disjunction not happening in anaphase 1. Disjunction didn't happen in anaphase 1. This pair went to the same cell. This disjunction not happening. Disjunction not happening. We use the term non-disjunction. Non-disjunction. During what? During meiosis first, specifically anaphase first, results in 47. In this case, 47. It could result in 48. It could result in 45, depending upon what type of gamete you are taking for fertilization. Such cases are called such flaws, such defects. Such defect of non-disjunction will result in genomic mutation, where the number of the chromosome will change. See, I have told you there will be numerical change in chromosomes. In nutshell, my dear students, you have to remember that non-disjunction of homologous pair of chromosomes during meiosis 1, specifically anaphase 1, results in faulty gametes. Gametes which are having more number of chromosomes or less number of chromosomes. Accordingly, this non-disjunction is responsible for genomic mutations, be it aneuploidy, or in case of euploidy you will see some other thing responsible, because you have in this case a complete set of chromosome getting increased. Okay, now let us go and understand further about the types of aneuploidy. So, aneuploidy, my dear students, is further of 2 types. Okay, aneuploidy is further of 2 types. Aneuploidy is of 2 types. Pay attention. We'll go to euploidy and polyploidy in a bit. So we'll take it right now in a bit. Let's first of all try and complete this aneuploidy. Aneuploidy further, it's of 2 types. See, very simple. Here you have increase in number of chromosomes, either one of you, or deletion in number of chromosomes, either one of you. So accordingly you will have either hyperploidy. You can guess now what is hyper. Hyper means hyper, more, and the second will be simple, hypoploidy. My dear students, now if somebody will tell you what is hyperploidy, what is hyperploidy, rather than me making you dependent on definitions, you can yourself think and be wise that hyperploidy is what? It's that aneuploidy in which you have addition, or you have what? Gain of one or few chromosomes. Yeah, that means either one chromosome is added, or two chromosomes are added, or three chromosomes are added. We use the word few, because for more we use the set, that is euploidy. Similarly, if somebody will ask you what is hypoploidy, okay, hypoploidy will be what now? You can guess yourself. Will it be gain or loss? It will be what? It will be loss of one or few chromosomes. So you don't need to be that rote learner in which you are given definitions and you are rote learning those definitions. No, try and think about the words. In biology, a lot of the meaning is hidden in the words, and if you follow some sequence, you automatically come and understand about the definitions also. So hyperploidy, you have gain of one of your chromosomes. In hypoploidy, you have loss of one of your chromosomes. Accordingly, my dear students, let us look at some examples, very beneficial. Say for example, there is an organism, a human being. Normally we have what condition of chromosomal set? We are diploid, we have 2 N chromosomes. This is diploid set. I hope you remember N is what? N represents haploid set. 2N represents diploid set. N represents 23 chromosomes, 2N represents 46 chromosomes. N represents 23 chromosomes, 2N represents 23 pairs, that is 46 chromosomes. So suppose normal organism is 2N, okay, this organism is having 1 chromosome more. So how will I represent that? 2N plus 1, gain of 1 chromosome. This 2N plus 1 condition is called trisomy. Very easy to think, trisomy. Trisomy is that condition of hyperploidy in which an individual has diploid set with an additional chromosome. Okay, this you will see happening in some diseases, so rather disorders, like you have Down syndrome. We have to study these syndromes ahead. You have Down syndrome. You have similarly Patau syndrome. You have similarly Edwards syndrome. Okay, similarly you have Jacobs syndrome. Okay, these are some of the syndromes, some of the genetic disorders in which somewhere, say for example in Down syndrome case, you will see that in chromosome number 21 you will have an additional set of this chromosome. In chromosome number 21 you will see one additional chromosome. In Patau syndrome you will see that there will be additional chromosome present in chromosome number 13 pair. In Edwards syndrome you will see there is additional chromosome present in chromosome number 18. So either chromosome number 21 pair is affected, this is having additional chromosome. Either chromosome number 13 pair is affected, it's having additional chromosome. Either chromosome number 18 is affected, it's having additional chromosome. Additional means what? So chromosome number 21 must be a pair in an organism, okay? This is normal chromosome number 21, a pair. But if you have one more chromosome number of 21 present, how many chromosomes are present in chromosome number 21 here? 3. This condition could be represented as 2N plus 1. You call it what? Trisomy. This is the condition with your Down syndrome. Okay. So under all these cases, you will see what? Additional chromosome. How many? 1. Either in chromosome number 21, then it's Down. In chromosome number 13, Patau. In chromosome number 18, if you have additional chromosome, then it's called Edwards syndrome. Similarly, my dear students, you will have something like this. Say for example, you have 2N. Suppose you will be thinking, is it possible? Is it like only one chromosome is added? No. Sometimes you will have 2 chromosomes being added. You have a condition called 2N plus 2. What you will call this condition? You will call it a tetrasomic condition. Okay. So this condition is called tetrasomic condition. Okay. This you will be able to see in case of superfemales. Superfemales, so example for this is superfemale. See, superfemales have what? 4X chromosomes. So autosomes are normal, but XX chromosomes are like this. Normally, females have XX condition, but these have XXXX 4X chromosomes. Condition is tetrasomic. It's hyperploidy. Organism is called superfemale. Similarly, you have these which are called rare superfemales. In them, you will see condition like this. They'll be having 5X chromosomes. So the condition will become what? Condition will become 2n plus 3. Okay. So it's like this. We'll call it pentasomic condition. Pentasomic condition. Pentasomic condition you will be able to see in case of, let me wrap this off. Pentasomic condition you will be able to see in rare females. So the condition is like X, 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 X and X. How many X's? 5. Isn't it 2n plus 3? Normally it must be 2 X chromosomes, but it's how much extra? 3. That's why we say 2n plus 3, pentasomic. These females are called rare superfemales. Sorry, it's superfemales. Rare superfemales. So in hyperploidy, you saw gain of one or few chromosomes. Some of the students have requested that I may help them in writing. So my dear students, you can take a pause right over here and you can write down this stuff. Okay, that's very good. Let us now go to, my dear students, don't be confused over here. Number 1 type we finished. Now let us now go to hypoploidy. Okay, let's understand about hypoploidy. I'll wrap off this. Now let me make you understand hypoploidy. So, hypoploidy again, you have loss of one or few chromosomes. This is very simple. Suppose I'll make a condition like this. 2n minus 1. Here it was 2n plus 1. Now you will say 2n minus 1. Loss of one chromosome. Condition will be called what? Condition will be called monosomy. You can remember like this. Mono because one chromosome was lost. Tri because 3 chromosomes you have. Mono because one is lost. This condition you will be able to see in Turner syndrome. It's very simple. We have to go into the details of these diseases once we will start genetic disorders. Now, Turner syndrome, in this case, a female is having only one X. Another X chromosome is missing. What is the genotype? X0. This female will not be a normal female because she is lacking one X chromosome. Condition is hypoploidy because one chromosome is lost. Representation is 2n minus 1. You can call this female a monosomic female because her one chromosome is lost. This condition is given the name Turner syndrome. I will teach you about other characteristics of this, main characteristics of such females which have Turner syndrome ahead once we will start genetic disorders. Similarly, we have this condition 2n minus 2. Can you think about it? What is this? 2 chromosomes are lost. 2 chromosomes are deleted. This condition is what? This condition is called nullisomy. Okay. Nullisomy. Nullisomy because 2 chromosomes are lost. Okay. Nullisomy, usually these organisms, these individuals, these person do not survive. They finally die. You can think why they die. They do not survive. They die. They do not survive. They die because 2 of their chromosomes, an entire pair is missing. They will not be able to perform their metabolic activities normally. So they usually die. So this was all about aneuploidy and its subtypes, hyperploidy and hypoploidy. In case of hyperploidy, you have monosomy and nullisomy. This is it. Now we are supposed to go to what? Euploidy. This is also very, interesting because the duration then exceeds. So that's why I will finish in this lecture numerical changes and you will have lecture number 3 for chromosomal aberrations. Okay, so I'll wrap off this because now I have to straight away go to euploidy. So now let us go to euploidy. So euploidy is also called polyploidy. Okay. Now in this case, what do you have, my dear students? You have increase or change in complete set of chromosomes. complete set of chromosomes is changed. So this stands, you have change in complete set of chromosomes. Think like this, if some organism is 2n diploid, it will become 3n, or it will become 4n, or it will become 5n. What is happening? An entire chromosomal set is increasing. That's why you call these changes what? Polyploidy or euploidy. Okay. Now, my dear students, think. You have further 3 types of this. Okay. Number 1 is Autopolyploidy. Number 2 is Allopolyploidy. 3rd is Autoallopolyploidy. Very interesting. Okay, so you have 3 types. Number 1, number 2, number 3. These are the 3 types of your euploidy. Now, my dear students, autopolyploidy, this is very simple. Here you will see that the genome of the same species is repeated. Let's first of all go to autopolyploidy. We'll finish like this, followed by this, followed by this. So autopolyploidy is that case where you will see what? You will see that the genome of only same species is repeated. So genome of same species is repeated. I'm talking about autopolyploidy. Okay, so genome of the same species is repeated means here you are not talking about 2 different species. Once you talk about autopolyploidy, here you are talking about 1 species. Okay, so can't we call it intra-specific polyploidy? To be very short, I'm using PP for polyploidy. Intraspecific, because you will find this polyploidy within a species. So usually you will find these polyploidy cases in plants. Around about 30,000 to 300,000 plants which are there in angiosperms. They are basically in nature, they are polyploids, and this polyploidy has some advantage also, which I will teach you one by one. You will come to understand about everything. Okay, so this is more pertaining to the botany aspect. So in case of autopolyploidy, genome of the same species is repeating. Okay, that's why you call it intraspecific polyploidy. Now let me make you understand. Suppose I represent a genome of a particular organism, or rather I'll say that a genomic set. Suppose I represent a genomic set by a letter A. So this A is basically representing what? It's representing N, that is the haploid set. Okay, this is the haploid set. Now, if an organism is having this condition, AA, what do you call this organism? Normal. But if the same organism will be having now 3 sets, see, genome set of same species, up to this is normal. But if the same species set is again repeated in an organism will have A. How many sets? 3 sets. Is it auto or allo? Auto. So we'll call this organism autotriploid. This is a triploidy case because this organism is having 3 sets of genome. 3 genomic sets. Okay. This is an autotriploid. You will see banana. Banana in nature, it's naturally an autotriploid. For that matter, even grapes are autotriploids. Now, suppose if an organism has this condition, what do you call it? How many sets it has? Tetraploid. So we'll call it autotetraploid. This is the case which you'll see in various varieties of tomato in nature. They are autotetraploid. So this is your autopolyploidy, where an individual organism is having multiple copies of its genome. Either 2 copies, 3 copies, maximum it can go to octoploidy, when an organism will have 8 sets. Beyond Autopolyploidy, these lethal genes increase in number, and the plant is not able to survive. So beyond Autopolyploidy, you will not see any organism. Till Autopolyploidy, you will see that you will have this genetic diversity present in case of plants. Now, in nature, we have seen that Autopolyploidy has some advantages. It has some advantages. You can think. Try and think in this manner. If an organism is having 2 sets of chromosomes, okay, when the number of the chromosome increases, can't we see the number of the gene is also increasing? Okay, number of gene is also increasing, isn't it? When the number of gene increases, can we say that the product of the gene will also increase? Product of gene will also increase. I mean to say the proteins. The end product of gene will also increase. That means the proteins. If the end product of the gene is increasing, can't we think that this plant will not be having a normal size? Its size may increase because at genetic level it is having more information for more proteins. Accordingly, you will see this plant having what? It will be having a large size because of its multiple copies of genomic sets, multiple chromosomes, many genes, many gene products. Its size will be more. It will not be having usual size. The size of its flowers, the size of its fruits, the size of its seeds, the size of its vegetative body, be it the root, stem, leaf. So entire vegetative body, that will be not small, that will be large. So economically, if we talk about agricultural thing, these plants will be able to give you less product or more product. They will be able to give you more products. They'll be giving you big fruits. They'll be giving you big flowers, big stems and roots and leaves. So they are showing some effect. This effect where they are producing large seeds, large flowers, large body parts. This effect is called the gas effect. This effect is called what? The gas effect. A giant body because of what? Autopolyploidy. Say for example, you have an autopolyploid like this. So a condition is like this, which I have already made you understand. A condition like AAA. That's a condition of what? Triploid, rather autotriploid to be specific. And the condition like AAAA. This is autotetraploid. Pay attention. See, this has something to do with the fertility of the plant. Okay, so in this case, think about banana. In this case, think about tomato. Think about this. Okay. Have you observed that banana do not produce seeds? They are seedless. Why? Let us understand. If this plant wants to perform what? If this plant wants to perform sexual reproduction, it has to make what? Gametes. And in order to make gametes, it has to perform what? Meiosis. Okay. So, can this perform meiosis? Think. During meiosis, what has to happen? What is very important is, isn't it important? Rather, pair of homologous chromosomes is important, because during meiosis, this pair of homologous pair of chromosome undergoes pairing, and then you have disjunction. If this pairing of chromosomes is not possible, meiosis is not possible. Think. Now, this is a set in which the pair is present. You have chromosome number 1 here, chromosome number 1 here. So they will perform chromosomal pairing, homologous pairing. But this set is not having its pairing partner. So this cannot perform homologous pairing. This set will perform homologous pairing, but this set is not having its pairing member, because you have total sets how many? 3. 2 will do pairing, 1 is left as such. Similar is the case, say for example, I'll take a case like this, A. How many sets are here? 5. These two will do pairing, these two will do pairing, this will remain unpaired. No possibility for homologous pairing. So can we say that those which have odd set of chromosomal sets, which have odd genome sets, like this AAA, in them we will see that pairing is not possible. When pairing of homologous chromosomes is not possible, is meiosis possible? Is it possible that they will perform a meiosis? Meiosis is not possible. When meiosis is not possible, is it possible that they will form gametes? They cannot produce gametes. When they cannot produce gametes, can they perform sexual reproduction? No. Can we call these organisms fertile? No. What are they? They are sterile. When they are sterile, what is the method for them to perform reproduction? Is only vegetative propagation. That means they will sometimes use their stem, they will use their root, they will use their leaf for reproduction. What is that reproduction called? Vegetative propagation. Now, when they cannot make gametes, when they cannot perform sexual reproduction, will there be any sort of pollen grain formed or egg formed? No. Will there be any seed? No. So they will be what? They will be seedless. This is very important as far as NEET is concerned. Look at this. Homologous pairing possible? Homologous pairing possible. So what is the number of the sets in this case? Even. What was the number of sets in which we have odd number of sets? They are seedless and sterile. But those autopolyploids which have even number of sets, see, can they perform, although they'll be having more chromosomes, but can they perform meiosis? Yes, they can perform, because homologous pairing is possible. Because homologous chromosome pairing is possible. Meiosis is possible. Can gametes be formed? Yes. Can sexual reproduction happen? Yes. Are they fertile? Yes. Will they produce seed? Yes. So some of the autopolyploids will produce seeds, those which have even number of chromosomal sets. Some will not produce seeds, those which have odd number of chromosomal sets. That's why your banana is seedless, whereas your tomato is having seeds. Okay, this was very important. Try and clarify your concepts at this particular stage. Okay, so sometimes, my dear students, it's not necessary that you will wait for nature to go with polyploidy. Naturally polyploidy will arise. Sometimes we can use certain chemicals ourselves. Okay, so we have certain chemicals also by the help of which we can induce polyploidy. Okay, we can induce polyploidy. So for that purpose we have a chemical called colchicine. Okay, colchicine is a chemical, it's a drug which we use to induce polyploidy. Okay, it's a chemical which is extracted from, it's extracted from a part which is called corum. I hope you remember this from class 11. You have different modifications of plants. So it's extracted from corum. Corum of which plant? It's extracted from corum of autumn crocus. Autumn crocus is the plant. We call it colchicum. Colchicum is the scientific name. The name of the plant is autumn crocus. From its corum we extract a chemical called colchicine. Chemically we call it colchicine. This we use to induce polyploidy. So if you will spray this chemical, if you introduce it to a cell will perform DNA replication. That means the copy of the DNA will be formed, but mitosis, that is the breakdown, the cleavage, the break of a cell into two different cells, mitosis will not take place. So replication will happen. You will have copies of DNA. So if you have 2 sets, you can convert that into 3 sets. If you have 3 sets, you can convert that into 4 sets. Means you can induce what? You can induce polyploidy by spraying which chemical? Colchicine, because this will promote the replication of DNA. You will get copies of DNA, but it will not allow cell to undergo division, mitosis. It will stop. So accordingly, you will have one cell with many copies of DNA. You will have a polyploid cell obtained by the help of colchicine. This is also asked in your NEET examinations, like which is the chemical which is used to chemically induce polyploidy in plants. Answer is colchicine. Okay, now let us go to allopolyploidy. This was all about autopolyploidy. So you need to have this information about gas effect. You need to have this information about why some autopolyploids are fertile, some are sterile. You need to have this information about how you can induce autopolyploidy. Okay, this is it. Now let us go to allopolyploidy. Okay. So can you tell me the alternative name? Can you tell me the alternative name for autopolyploidy? It was intraspecific polyploidy. Okay. It's also called interspecific. What is the information you get from this word interspecific polyploidy? Here you will have two different species involved. So it's not going to remain confined to one species. It's going to be dependent on how many species? Two. So in this case you will see it involves what does it involve? It involves hybridization of two species. That's the logic. It's same. You will see more copies of a genomic set. But in this case, you will see an organism which has genome of two different species. That means we are talking about what? Hybridization. Let us get an idea. In this case, you will be having, suppose first of all, you will be having species 1. Let us take species 1 and let us take species 2. Okay. Species 1 organism, let me take its genome as AA and let me take this genome as BB. That means both of them are diploid. So how can we make the difference between the genomes? I have represented this by BB, this by AA. So this is one species, this is another species. Now let us take the gamete from this species. We'll be having only one because gametes are haploid. We'll be having only one set, gametes are haploid. Okay. Now let us do a kind of cross hybridization. So under artificial circumstances, we are self-pollinating this plant and we are getting a hybrid. We will get a hybrid which will be having A set and B set. So is it a normal variety or a hybrid variety? It's a hybrid variety because it has got genome from two different species. Okay. Normally, my dear students, this hybrid will be sterile. Because, think why it will be sterile. This chromosome set, it will not pair with this chromosome set because they are belonging to two different species. So hybrid itself will not be able to perform meiosis because in it pairing is not possible between chromosomes because the sets are different. Okay, although it is diploid organism, although it's having two sets, but the two sets belong to two different species. They will not perform homologous pairing. Now, if to the same hybrid, now what will happen? Now in nature they undergo what? Polyploidy. That means the same case, the cell will perform DNA replication, but it will not let what? Mitosis to happen. This cell will then be converted into something like AA because it will be replicated, BB. Okay, either this can happen naturally or it can be induced by what? It will be induced by this chemical called colchicine because I have told you colchicine induces DNA replication. Colchicine starts replication. It stops mitosis. So once if you will use this cell and you will apply colchicine to it, this will be converted into a cell which will be having two sets of A, two sets of B in the same cell. This is again what? Isn't it polyploidy? Again, you have 4 sets of genomes. So this is what? Tetraploidy. But in this case, you have how many species involved? Genome of how many species involved? 2. Species 1, species 2. That's why you call it allotetraploid. Okay, that's why you call it allotetraploidy. And you call this plant what? You will call it an allotetraploid. Pay attention. Now, you have certain allopolyploids which are generated in agriculture. Let me give you example for them. You might have heard about triticale. Triticale is an example of an allo. It's a hexaploid. Okay, it's a hexaploid. Its genomic condition is something like AA, BB, XX. So it has how many sets of genomes? 3. Okay, A, B, and X. It's a hexaploid. It's obtained by doing a cross between. It's not a normal serial. It's an artificial serial. Remember this also. It's an artificial serial which has been produced by doing a cross between wheat, which is natural wheat, and rye. When a cross is done between wheat and rye, a cross was done between wheat and rye, and the species and this plant triticale was obtained, which was a hexaploid. Similarly, there is this Raphanobrassica. Raphanobrassica, which you call rabbage. It's a plant which has half of the characteristics of radish and half of the characteristics of a cabbage. This plant has been obtained by doing a cross between radish and cabbage. So you have 2 different species, radish and cabbage, which has resulted in the formation of this kind of plant, which is called Raphanobrassica, which has a characteristics of both radish as well as cabbage. So it has got a root like radish and it has got this leafy part like cabbage. This was obtained, this was actually produced by a scientist who is called Karpchenko. Karpchenko did this Allopolyploidy experiment where he produces Raphanobrassica. Let me give you the details of this Raphanobrassica because this is very interesting. Let me show you. So you might have observed that all of these things, polyploidies, whether auto, allo or autoallo, they all happen to the plants. Okay, so let us talk more about this Raphanobrassica. What is the common name? It's not cabbage, it's rabbage. So who produced this plant? The name of the scientist is Karpchenko. Okay, Karpchenko. So what did Karpchenko do? He took radish and he took cabbage. What are the scientific names of radish and cabbage? It is Raphanus sativus, and he took cabbage, which is Brassica napus. Okay, so we have radish. This is the experiment which was done by Karpchenko, and cabbage, Brassica napus and sativus. Both of them are diploid organisms. Okay, this is diploid and this is diploid. Both of them have 18 chromosomes. Now, let us, for simplicity purposes, let us represent the genome of radish by RR, because it's diploid, I have told you, and let us represent the genome of cabbage by CC. Now let us do the experiment which was done by Karpchenko. Let's obtain the gamete over here. Gamete is R, only one type of gamete, and gamete is C. Okay. Now let us do the cross between these. Let us hybridize them. You will get a hybrid, which is having this combination of R and C. Okay. Now what Karpchenko did afterwards, he used He treated this with colchicine, so he gave it a colchicine treatment. After colchicine treatment, genome became RRCC, means 2 sets of genome from Raphanobrassica, Raphanus sativus, and 2 sets of genome from Brassica napus, the cabbage. This was given the name because this plant had root features which were similar to the radish, and its shoot resembled that of the leafy part of a cabbage. So this newly formed artificial vegetable is given the name Raphanobrassica, which was produced by doing an experiment of Allopolyploidy, because in this case you also see that 2 species, genome of 2 species is involved, and that too this genome is present in polyploidy because it is having extra copies, and this is the case of an allotetraploid because total sets are 4. So this is the case of, it's not a diploid, haploid, triploid, it's an allotetraploid. So Raphanobrassica is an example of an allotetraploid which was produced by Karpachenko by doing a cross between Raphanus sativus, okay, and Brassica napus. Let us take the last type of polyploidy, that is Autoallopolyploidy. This is very simple. So till now we are clear with Autopolyploidy, we are clear with Allopolyploidy. This was the first type, this is the second type, and now we are taking the third type, that is Autoallopolyploidy. Autoallopolyploidy is, my dear students, a case in which you will see that 2 species are involved, hybridization is present, and at least you will find out there is 1 genome which is present in more than diploid state. Let me show you first of all the genomic condition. The genomic condition will be somewhat like this. You will have, suppose, 4 copies of A set is present in 4 copies, and you have 2 copies of B. So what is the condition actually over here? You have 4A and 3B. The organism is what? Is it diploid, triploid, tetraploid, pentaploid, or hexaploid? It's hexaploid, okay? This condition you will see in one of the varieties of sunflower, which is called Helianthus tuberosus. Okay, Helianthus tuberosus is naturally having autoallopolyploid condition. So condition of genomic level is this. Now what is the condition a plant has to fulfill in order to be called as autoallopolyploid? Number 1, it must have more than 1 genome. Okay, so more than 1 genome is present in case of autoallopolyploid. Now this is the case with autopolyploidy also. This is the case with allopolyploidy also. You must have more than 1 genome. Okay, so you have more than 1 genome. See, you have A genome, you have B genome, and A genome you have in 4 copies, B genome you have in 2 copies. Okay, similarly the another criteria is that at least 1 genome. So at least 1 genome must be present in more than diploid state. This is the condition. Out of the 2 genomes, you must have at least 1 genome present in more than diploid state. So genome B is present in diploid state, and in this case you have genome A present in more than diploid state. It's present in 4 copies. So once your plant fulfills these 2 criteria, you will place this plant under the class of autoallopolyploidy. So in case of autoallopolyploidy, you must have more than 1 genome, 1 condition, and the other genome, at least 1 genome must be present in this plant in which you will find more than diploid set. So you will be having more than diploid state with at least 1 genome. If this is the criteria, it's called autoallopolyploidy. This is all for today, and do try and explore this video as much as you can. If you have any questions, so drop that question in the comment section below. I'll answer your query. In the meantime, don't forget to like, share and subscribe my channel so that I can reach to more and more students. Even we have now one part left, that is called chromosomal aberrations. We'll complete that in the next video. Till then, take care of yourselves. Stay home, stay safe. Fi Amanillah.